Hello, welcome to the new session of Premade Pro, and we are going to continue with the chapter Molecular Basis of Inheritance, and with the topic of transcription. I have ended up to a portion of transcription yesterday, and today we will start from the next part. Okay, transcription is a process. Transcription is the process of changing of DNA to RNA. When the DNA is converted into RNA, the process is called as transcription, and when the RNA is converted into an enzyme or protein, it's called the translation. Okay, this total feature is called the central dogma. Okay, now give the heading as transcription. Transcription. Okay, so yesterday I have already said transcription, that is the mRNA synthesis from the 5' end to the 3' end and the addition of the ribonucleotides. Okay, so today when don't you think a question arises from it? Why do you think that both the strands of the DNA do not participate in the process of transcription? Both the DNA, if they get participated in the transcription, they will code for different amino acids. Okay, so the total RNA that will be produced after transcription will haphazard the total central dogma. That is why a single strand of the DNA participate in the transcription while the other strand acts as a template. Okay, so why both strand do not participate in transcription? Why both the strands do not participate in transcription? Okay, so this is the upper strand, and this is the lower strand, upper strand and lower strand. Let it contains A, U, G. Okay, and this contain, okay, this is the upper strand and this is the lower strand. Okay, now here is G and C. Okay, here what happens? Both mRNA sequence are complementary. Okay, so they form H2 bond, hydrogen bond. As both the strands are complementary, they form the hydrogen bond. Now what happens? mRNA becomes double stranded. mRNA becomes double stranded. So no tRNA binding happens. So tRNA binding never happens. So no tRNA binding happens. What it makes? If the tRNA doesn't bind, so what it means? No protein is synthesized. So if protein is not synthesized, then there is no need of transcription and translation. There is no need of transcription or translation, because the body actually works on the policy of the enzymes. Enzymes means proteins. Okay, so codon of both RNA are different. Codon of both RNA and RNA must have to be different. Now what happens? So they bring different. So they bring different amino acid. So two different type of protein. So two different type of protein. So it will complicate the machinery. Two different type of protein from the same amino acid ultimately will complex the machinery. The machinery will not be able to understand why they are making it from these two different amino acid. See, the AUG codes for methionine. Now if AUG codes for methionine as well as the valine, so there will be a systematic error. Okay, so there will be a systematic error in the machinery. So they will complicate the machinery. Now what happens? Now the next part, concept 3, what we will write is the transcriptional unit. Give the heading as transcriptional unit. Transcriptional unit. Transcriptional unit. So transcriptional unit, I need to draw it. Okay, now okay, these are the transcriptional unit. Now what happens? Let me draw it first. Okay, now bond, so this is the 3' end. So this is the 3' end. This will be the 5' end, and here this is the 3' end. This will be the 5' end. Okay. Now what happens? This is called the transcriptional unit. This total is transcriptional unit. Okay. So, and this region, let this region is called the promoter region. Okay, this red region is called the promoter region. Okay, this white is called the structural region. This white is the structural region. and this is the terminator region. Terminator region. Terminator means stop. Transcription stops here. Transcription stop. Promoter region means transcription starts. Transcription starts. Okay. So now take it as a note. Formation of mRNA. Formation of mRNA starts from structural gene / cistron. Okay, this is the case number 1. Case 1. 3' to 5' template, and 5' to 3' it is non-template. Okay, so this is the PR, this is the TR. PR means promoter, TR means terminator. Okay, now mRNA show antiparallel with DNA. mRNA always shows the antiparallel nature with DNA. mRNA shows antiparallel with DNA. Okay, now 3' end of mRNA, towards terminator. The 3' end is always towards the terminator end. Okay, now case number 2. Don't, case number 2 is not required for it. So now transcriptional unit. Okay, see, there is the code is made up of RNA polymerase. The code is made up of RNA polymerase. Okay, it is a core enzyme. Okay, now what happens? RNA polymerase is a core enzyme, binds to the promoter. RNA polymerase always binds to the promoter, with the help of sigma factor. This factor is asked in the NEET. This factor. Okay, sigma factor is a protein, is also protein. Okay, now RNA polymerase, that is the core enzyme, binds to promoter region. RNA polymerase Binds to the sequence. RNA polymerase binds to the particular sequence. Binds to particular sequence and just write the notes with me. It will help you to memorize it, because writing once will be equal to the 4 to 5 times of studying. Okay. Particular sequence in PR called consequence sequence. There is a called a Tata box. Okay. Tata box. It is also called a Pribno box. And TTGACA, there it will be upstream process towards the promoter end, as well +1 the downstream process towards the terminator end, that is PR end means 3' end, TR end, terminator end, that is 3' end. Okay. If sigma factor absent, is absent, then the transcription occur non-specifically. Rho factor protein actually binds to code enzyme and recognize the terminator. Okay. Next we need to start another it's 18-minute video already. Okay, so let's give a brief idea of the transcription in the prokaryotes. I'll end up with that, and we'll continue with the transcription in the prokaryotes in the next video. So a brief description in prokaryotes, give the heading as prokaryotes. Okay. Only one type of RNA polymerase. So prokaryote contains only one type of RNA polymerase. One type means for mRNA it's PROYIDE codon. You will not get these questions anywhere. This information are only given by me. I can claim it. Okay. tRNA is given by carry amino acid. Provide codon carry amino acid. While rRNA in ribosome. all type of RNA is needed for protein synthesis. All type of RNA is needed for protein synthesis. Okay, all the type of RNA is needed for protein synthesis. Now what happens? Let valine with MET valine. Okay, cytosine, here also it is cytosine. This is the 5' end, this is the 3' end. Okay, now rRNA catalytic role, structural role. Okay, it helps in binding of mRNA to ribosome, Okay, so the central dogma is DNA to RNA and to protein. DNA to RNA is called transcription. RNA to protein is called translation. Okay, so in the next lecture I will start from the transcription in prokaryotes. How the transcription goes on in prokaryotes will be continued from the next video. Okay, till then, good night, and please remember to subscribe this channel, and if you are new, and please remember to read the whole video. Probably you are getting something difficult to understand. If it's there, please write it in the comment box, because I do not get interaction with you except with the comment box. Okay, so if you find any problem, write it in the comment box. I will definitely try to solve it. Okay, and next, please share this video with your friends, those who are giving the NEET, and please like it. So good night.